Hi, everyone. Good to be with you today. Today, as our guest, we have Lawrence Bosman, who received his doctorate in developmental neuroscience at Amsterdam, then a postdoc in Munich, and followed by a postdoc at Rotterdam with Chris de Zoo, where he is now a faculty. And Lawrence is it's so good to have you with us today, and I know that you are going to talk about learning and changes that take place in the sensory representations in the cerebellum. So welcome, and thank you.
So thank you very much, Reza, for first of all for organizing this very interesting series of lectures, and thanks for inviting also me to take part in this. I'm very happy that I can share some of our work on what I think is the importance of context for understanding cerebellar activity. And I wanted to illustrate that with a little video from our German neighbors, where you can see that context can be quite relevant, even though you have a very skilled movement, if it's out of the sensory-motor context, it can still completely fail its goal. Yeah, and I would like to get away from soccer and get into the cerebellum, and use the following 4 questions to discuss the impact, what I think of context on cerebellar function. And it starts with sensory information, and that's, as you can see, the biggest part of the talk, and then mainly related to climbing fibers. So first of all, how reliable actually is sensory representation in the cerebellum, and is there a role for rhythmicity? We'll get back to this, and how stable or plastic can this sensory representation be? And after going through all this, also look at the motor output that is ultimately the goal of sensory processing. So for this, I will mainly look at Purkinje cells. I have here this beautiful image from Wei-Terry Geer's lab, where you can see a Purkinje cell, cerebellar Purkinje cell, with the climbing fiber really enwrapping the primary dendrites. And when that's active, that generates a huge complex spike that is relatively rare and embedded in a context of simple spikes. So when I entered the lab of Chris already quite some time ago, together with Bas Koekoek, we set up a recording setup to record from Purkinje cells in at that time still anesthetized mice while we were stimulating the whiskers. And when we did that, we found that there are quite a few cells that show this very clear complex spike response every time, or actually not every time. Even in this case, it's only about 50% of the stimuli in which we could trigger a complex spike. So in this sense, we could think, indeed, complex spikes in Purkinje cells can be quite a reliable representation of sensory input. And they can do that because of a number of reasons. So there is a direct connection from the trigeminal nuclei, where the sensory input from the whiskers enters the brain, to the inferior olive, which is the source of the climbing fibers that you saw in the previous picture, and that whose activity triggers the complex spikes. But there are also subcortical loops that can then further, so for instance via the NVJ or via the cerebellar nuclei, also affect inferior olivary activity. And there's also then a feedback loop, and this of course is also related to cortical loops, where the cerebellum projects to the thalamus and then to sensory and motor cortices back to the inferior olive, or via the pontine nuclei in the mossy fiber pathway, affecting directly the simple spikes. And this is then so the simple version of whisker control, where input can go both directly to the cerebellum or can be digested first by the thalamocortical system and then as descending input coming to the cerebellum. And the cerebellum can, at multiple levels, also affect the motor outputs. So sensory stimulation can trigger complex spikes, but what does this mean for the mouse? And from this, yeah, I borrowed a few slides from Reza's work. I think it's a beautiful paper, David Hertzfeld and Reza in monkeys, where monkeys are invited to make or trained to make a sagat, and then sneakily in some trials, the target area is moved, and there is a misrepresentation between the expectance and the actual observation, and this can then relate to the firing of complex spikes. And then they could show, I don't know whether you can see my mouse, but I think it's obvious anyway. These complex spikes relate also to the simple spikes in an angle-specific way, and they could show that in the absence of complex spikes, also the simple spike modulation is gone. And yeah, their story doesn't end here. And also together with work from Stephen Lisberger, we know that this can be also a mechanism of learning where the complex spikes gather sensory information and help the cerebellum adjust to changing environments. But that goes a bit fast because I'm still in my mind at the first initial sensory representation. So we went back to the whiskers, now together with Qian Yu and Tycho Hoogland, who did two-photon imaging, this time in awake mice, and they gave air puffs to the whiskers, and also under this condition in awake mice, we could see that this could trigger reliable sensory responses. And then we started to wonder, yeah, to what extent is this now also a selective representation? So an air puff also makes a lot of sound, and is quite a strong stimulus. So could we, with giving more localized stimuli also discriminate, for instance, between different areas of the face? So Chiang used a filament to touch the head of the mouse at 4 different locations: the whisker pad, the cheek behind the whisker pad, the upper and the lower lips, and also displayed sound and gave visual stimuli. And in the lower panel, you can see from a single neuron, a single Purkinje cell, complex spike responses to all these stimuli. And there are endless discussions started as to say, yeah, which cell, which histogram presents here a sensory response. Right? So the LED, so the visual stimulus clearly evoked a response. And so the gray lines indicate so the 67, 95, and 99 percentiles of shuffle data. So when it exceeds the upper gray level, you could say it's less than a 1% chance that is a random spike. But none of these are so nice looking as the cells I showed you in the beginning. And then, well, I said, well, let's make a histogram of all strengths. And then I expected to see a bimodal representation with some cells not responding, others are clearly responding. And to, at least to my surprise, we found if we plot on the x-axis the distribution of all sensory representations in CRUS1 to tactile, auditory, and sensory responses, we found a sort of gradual distribution where obviously cells with a Z-score of more than 12 are highly, highly responsive, and cells with a Z-score of 0 are not responsive at all, and all flavors are in between. So it was principally not possible to say in an objective way, well, this cell is responsive and that cell is not. There's a gray zone, and actually quite a lot of these cells that every now and then fire a complex spike in response to sensory stimulation, but often do not. And yeah, then we even had to convince ourselves and the reviewers that that our representation was selective, and we did this by plotting the average strength per cell versus the strongest response, and there's only very weak correlation. Right? So also its principal component analysis was just above chance level. That much of the responses, much of the tactile responses could be evoked by any stimulus location, but not completely. So there is still a sort of somatotopic representation in at least the part of CRUS1 where we've been recording from, and not just a sort of alarm signal, something is happening. But the separation between that seems to be quite subtle. So sensory representation in the cerebellum and all our data come from CRUS1 and CRUS2, and of course I'm aware that cerebellum is much bigger than that, but in CRUS1 and CRUS2, sensory representation can be quite reliable, but typically is not so. In some paradigms, and in Gesa's work, for instance, there's a clear role for complex firing, which is not so obvious in our case, but I'll come back to that later. And then, yeah, so as said, we started this in the context of whisker movements and whisker touch, and whiskers are, of course, rhythmic in their activity. So we were also wondering to what extent can there also be rhythmic representation in the cerebellum, and especially also in the complex spike response. And that is not such a weird question, as there's, as I said, a direct connection from the trigeminal nucleus to the inferior olive, which is known to be a rhythmic area. So please don't pay too much attention to the spoiler alert yet. Because so first I would like to show this work from Yoshi Jerom's lab with imaging. They could show that there are subthreshold oscillations. They're by far not the only group that demonstrated these subthreshold oscillations. But I think this is a really nice representation for this. In tissue slices, when not much is going on, there is a rhythmic activity in the inferior olive, and as the inferior olive sends the climbing fibers to the Purkinje cells, these must also be in the complex spike responses. Now, together with Mario, actually, Mario Nigrella did it, took this part for his, and he could, in a computer model, in a simulation of a network of the inferior olive, could show that these subthreshold oscillations can also be readily evoked when adjusting the correct ionic currents. And by the gap junctions that are very prevalent in the inferior olive, these can be synchronized between cells. Without gap junctions, the oscillations are still there, but they lack their synchrony. And if Mario made then a model stimulus, you could see this interesting observation that upon the stimulus, in around half or 1/3 of the trials, there was a spike that would obviously then trigger a complex spike in the Purkinje cell if this would be a real brain. But also in a quarter of the trials, when there was not a spike on the stimulus, there was a spike later on, and this spike later on coincided with the occurrence of a secondary spike in the trials that had a initial fast response. So there seemed to be a window of opportunity for the complex spikes, either to fire early on or at the second period of the subthreshold oscillation, which you can also see reflected here in the histogram. And when we compared that to mouse data, we showed a similar thing. Of course, now there's a latency because the signal has to come from the whiskers to the cerebellum, so that takes a bit of time. But for the rest, the principle is the same thing. In the blue circles, one can see the first response, and the pluses are the secondary response. So in trials where there was not an early spike, there was often a spike in the second wave. So it seems that our model and our real mice for once confirmed each other, and showing that there can indeed be rhythmicity in sensory representation. Then we argued that if there is a sensory component of a rhythmic component in sensory representation, there should also be resonant frequencies where you could easier evoke a complex spike response than others. And in general, if one would increase the frequency of the whisker stimulation, we would also expect a higher rate in complex spike frequency. But no, whatever we tried, we could not elevate the complex spike response over the experiment. Right? So at different frequencies, and we used different types of rhythmic frequencies, gallop frequencies, combinations of different frequencies. Whatever we did, we could get responses and even strong complex spike responses, but we could not crank up the frequency of the complex spike responses. And then to explain that, we went back to the two photon data. And you see here the combined data of a single field of view, which had around 16 Purkinje cells. And the browner the color means the more cells were active in a given frame. And these were recorded during air puff stimulation, whisker pad, upper lip, lower lip, and cheek. Obviously, in this recording, in this area was really sensitive towards the air puff recordings, a bit towards the whisker pad, which was more subtle stimulus, but not to the upper lip, lower lip, and cheek stimuli. And then, there are two things that strikes us. So first of all, it seems that the air puff can generate many more complex spikes. But if we then look at the total amount of complex spikes, it turns out that air puff cannot recruit any single complex spike. And the complex spikes that we see during the response actually come at the expense of the intertrial firing. So what we see is a reshuffling in time of the complex spikes and not a recruitment of more complex spikes. It's, of course, it's always only true for our paradigm in CRIS1, but it seemed that the complex spikes are really aiming to be around 1 Hertz and can shuffle a bit in time, but over longer periods they stay constant, even though there's a lot of sensory representation going on. And the second thing we observed is that the pins with the darker colors, so the more simultaneous complex spikes, they are mainly organized in sensory response, meaning that in the intertrial intervals, it was mainly occasional cells that had a complex spike, but no synchrony, or synchrony is maybe with this time resolution, not the right word, but not well timed over different areas, different Purkinje cells. And yeah, we think this could have a homeostatic function. Right? As complex spikes are really important for learning, you should also not completely lack them. That would also be a learning signal. So, to summarize this part, in specific behavioral context, complex spike can account errors, and these are then unpredicted events or outcomes or mismatches in the outcomes related to the prediction. Complex spikes can represent sensory inputs, but the specificity can be highly questionable, and it's also doubtful whether they are really reliable in many cells. And I think it's still fascinating that so many cells do represent sometimes a complex spike, but not all the times. And overall, it seems that there is a strong tendency to be homeostatic, right? Not to increase the firing too much, not to deviate too much, but that there is a temporal reordering of the complex spikes. And then if they come together, it could be a salient signal, whereas when they are isolated in single cells, they're probably, well, at least that would what I would hypothesize, they're probably there to keep the calcium concentration at a sort of baseline level, not to trigger plasticity. Nothing has to be learned. So there is some rhythmicity in the sensory representation that we could find, but only over a very short time scale, so 200 milliseconds. Then there can still be an effect of the subthreshold oscillations over longer times. Sensory input to the synaptic input to the inferior olive overrides the subthreshold oscillations. And that brings us then to the third question: to what extent is this representation stable? And for this, I would like to show work that was largely done by Lorenzo Pina, a PhD student in our lab, and he adapted a association paradigm that was pioneered by David Kleinfeld and later further developed in Svoboda's lab, in which mice were trained to associate the position of the insertion of a pole into the whisker field with a reward. And when the pole was raised outside the whisker field, it was a no-go signal. So, and then if the mouse correctly recognized the position of the pole and he would lick within a defined reward window, he would get a water reward. The way we constructed our setup implied that the rising of the pole was associated with a clearly audible sound. And when we looked over training at our mice, we noticed that obviously naive mice wouldn't lick, which is quite logical. They didn't know that there is water in the setup, so why would they lick? And then over time, they got better and better in discriminating between go trials and no-go trials, and in timing their response. Now, for this experiment, it's important to realize that our mice had to trigger the water reward. So they had to first make a response that triggered the water reward. There's later that were simultaneously, there have been very nice studies, for instance, by Kurt Ho, but also by others who gave this a similar paradigm, but then as an associative paradigm, where a reward was always triggered with the present, the stimulus was always triggered by the presentation of a reward, which was not in our case. So in our case, the mice had to do something before they got the reward. If we then recorded, and we'll soon go to the data, the complex spikes in the Eve mice, in sort of half-trained mice and in trained mice, we noticed that there were many complex spikes initially triggered by the sound, which marked the onset of the trial, but it was identical in go trials and no-go trials. So it didn't have any salience for the mouse, other than sort of wake-up call from, look, now the trial is going to start, but it could not predict whether there would be a reward or not. And then we noticed that during training, we saw the complex spikes that were triggered by the sound disappear, and instead see an emergence of a second peak of complex spike that were triggered by the touch, which was initially basically ignored. And in really good mice that made little mistakes, there was little response to the sound, but a clear to the touch. At the same time, simple spikes also shifted from a relatively early peak to a later peak. So now in 3 real trials where we recorded extracellularly from Purkinje cells, either in naive mice, in mice that were not so good in training, and mice that were really good. And then you can see is these green dots of the 30 trials. You can see that they indeed respond initially to the sound, and later on there was more of a response. You can see that here at the end of the sound, so at the moment when they could feel the whisker bar. And so comparing go and no-go trials, obviously the naive mice had no clue that there was a difference between them, so their responses were similar. And then in non-expert mice, the tactile cue was stronger in the go trials, where there was actually touch, than in the no-go trials, although there was some sort of a resonance still also visible there. And then in the expert mice, the first peak was much less, and the second peak was much bigger. Now, these were done with electrophysiology, and our setup, we can unfortunately not record over different days. So for that, we went into a mini scope together with Tycho Hoogland, and we could record during the whole training. And based on the location of the cells, we are pretty confident that we can follow individual neurons throughout the training. And you can see here an example of learning, but the important thing here is in panel F, where we show for an individual dendrite at day 4, so early on in training, day 13 and day 20, that in day 4, early on in training, we had only a single peak, a double peak, halfway the training, and a single peak at the end of the training. So it's actually individual Purkinje cells that change from an auditory representation to a tactile representation. The auditory representation is not so weird. The sound is quite overwhelming for the mouse, and you see it doesn't know in which context it is. But in the end, the mouse realizes that, I don't know whether that's consciously or subconsciously, but at least the mouse realizes that that is actually the touch, which initially seems to be quite subtle. The touch is the salient stimulus, because that is a predictor of whether a reward can be obtained or not. So during training, and this is again in quiz 1, the sensory modality represented by the complex spike of individual Purkinje cells is subject to change, right? And so depending on the context, cells that were initially responding to sound become responsive to touch. When we looked in at the temporal correlation, so you see here stacked line plots. So every line here represents a mouse, and the top line is the average. Here again for the complex spikes and simple spikes. You can see that there is first a relatively small peak in trained mice. That is the sound induced response, and a broader, bigger peak, the touch-induced response. And then the simple spikes, they come up here related to the licking. But of course, the mouse doesn't lick in every trial the same latency. So we plotted this again. This is the very same data, but then aligned on the first lick. And yeah, a bit to our surprise, we found that there is no correlation between the complex spike timing and the licking. So the complex spikes seem to guide the behavior. I will come back to that, I think, in the next slide. They seem to guide the behavior whether to lick or not to lick, because these are expert mice that make correct decisions. But on a single trial level, there is not a fixed latency between the complex spike and the lick. So it's unlikely to be the motor signal. It's more like a sort of permissive signal from now this is the context to lick, rather than to an actual signal to the tongue. But in that respect, the simple spikes seem to be more related. And yeah, of course, the licks are detected at the maximal protrusion of the tongue, so the tongue always starts before that, but probably still after the increase in simple spikes. Now, that is, of course, nice. At least I think it's nice that the cerebellum is involved in this task, but is it also required for this task? And we answered that by doing the same training in mice that lacked PP2B or PP3R1, which is required for the insertion of AMPA receptors, and its absence reduces the ability of Purkinje cells to express LTP, long-term potentiation. And in these Purkinje cell specific deficient mice, you can see that at the end of the training, they reached a similar level of hit trials, but they needed a longer time to learn the task. And so it's not that without potentiation in the cerebellum, mice could not learn, but they were not so efficient in learning as the wild-type littermates were. And then I think that's another, at least to me, surprising observation that we made, is that if we compare then the hit trials in trained wild-type and trained mutant mice, so as I said, in the end, even the mutant mice are quite good. And in terms of kinetics of their licking, we could not discriminate them from the wild-type mice. Also not in the timing, right? So we took here only the trials where he licked during GO trials and not early licks. If we compare that to the black ones are the wild types, the red ones are the mutants, there was not that much difference in the complex spike, which is also not expected as it's not a mutant of the inferior olive. But whereas the wild type mice typically solve this task by increasing their simple spikes, the mutants actually did so by decreasing their simple spikes. So apparently there are two ways to solve this task. One is to, via potentiation, get an increase in simple spikes. And the other way is to get a decrease. Well, LTD is not deficient in these mice. Get a decrease in simple spikes. But then these data are also from the leaks, the complex spikes and the simple spikes are all from the same trials. So it means that also the increase of simple spikes that we show here in the wild-type mice is not necessary to get the licking response, because the mutant mice with a decrease in the simple spikes can get the very same behavioral response. Well, it takes longer to get them well timed and at the right trials, but eventually the motor output is not different, although they have the opposite modulation of their simple spikes. So it seems that in this task, neither the complex spikes nor the simple spikes are necessary for the actual motor execution, but are required for the proper association of the sensory context and the motor response. Yeah, so we had same action, but different encoding in terms of simple spikes. So from this, we concluded that complex spikes in CRUS1 can relate to the salience of events, right? So they are not direct sensory representations because the sound and the touch are identical at the beginning and the end of training, but the context is different, and that is reflected in how they respond. Massive complex spike firing can trigger simple spike plasticity, and that's required for cerebellar learning. That's a little bit outside of what I've shown here, but many papers are in line with that. So from Brian's or Chiang-Yu's work, we concluded that sporadic complex spike firing, so in single cells, not in groups of cells, not in ensembles or microzones, it's probably important for homeostasis. Simple spikes relate to motor output, but both complex and spikes and simple spikes are not essential for the motor commands themselves. yeah, so many people studied these studies in context of cerebral cortical activation. Like in Mitchell Diamond's review here, the cerebellum even has a different color than the rest of the brain, and seems to be completely disconnected. But we do think that it is important also for affecting what's going on there. And it can do so. Manuel Novella summarized this in a systematic review that was published 3 weeks ago, where all the outputs of the cerebellum go. And as you can see, these are all the direct output, not to the spinal cord, but to all the other areas. These are really widespread, and in line with, with our initial diagram, that this is quite a complex network. And then, I would like to conclude with two slides on the motor output. And this is work that was done with mainly by Vincenzo Romano, who was at that time a PhD student and now has his own group at our department. We were inspired by a paradigm from Egidio D'Angelo, who was also a collaborator on this project, where he showed that, or actually his in vitro data suggested that a brief period of high frequency or 4 Hertz input to the granular cell layer could trigger plasticity. So what we did, or what Vincenzo did, is that after a period of a low frequency air puff stimulation of the whiskers, there was a 20-second 4 Hertz stimulation of the whiskers, and then the post-induction phase. If we looked then again, we stacked line plots to the whisker movements, you see that before and after there was a change of about 2 degrees more protraction after induction. That's not a huge effect, but it was really sustained, and we could very reproducibly see it in all our mice. So after the initial retraction, which was caused by the air puff, the mice make a reflexive protraction, and this reflection protraction is stronger after induction. And you can see it here on average, it's like 1.8 degrees more protraction. The complex spike responses didn't change, not in any way systematically before or after induction, but the simple spikes, and especially here shortly after the air puff, they are now increased. So we see both an increase in simple spike firing and a further protraction of the whiskers. And what Vincenzo then did is to relate the variance between each trial and show that indeed an increase in simple spike typically corresponds with a stronger protection and along the 45 degrees line. So that means that there is a simultaneous increase in simple spikes and protraction. We found the best correlation. So this suggests, and you can almost see it here in the shape of the modulation of the simple spikes and that of the whiskers, there seems to be a sort of representation of the ongoing movement in the unperturbed situation. But after stimulation, we found that there is an increase above the 45 degrees lines, meaning that the simple spikes, and you can see that here, now precede the movement. So they seem to switch from a more or less a representation of the ongoing movement to a more motor adaptation function where they actually can trigger the further protraction. Vincenzo could show in a mutant mouse, again this PP2B mouse, the cerebellar mutant mouse, that if you lack the ability to show parallel fiber LTP, then also this change in timing and increase in whisker protraction is gone. So this is really mediated, this increase is mediated at least also by the Purkinje cells. And together with Evan Reddington, we then looked and Vincenzo again, we then looked at the correlation between simple spike firing and respiration. And we found that at rest, we found a similar thing as at rest in the whisker reflex, that the modulation of the simple spikes, at least in a large proportion of cells in CRUS1, follows that of the breathing, which is in dark, in light blue. However, when we gave an air puff to the snout of the whiskers, snout of the mice, this triggered inspiration, and this inspiration was preceded by an increase in simple spikes. So just as in the whisker situation, we saw here that in the unperturbed situation, there seems to be a different correlation between simple spike firing and motor output than when something unexpected happens. And then simple spike can precede their firing and modulate the motor response. And again, here we could show in a mutant mouse that you need LTP of the parallel fibers to mediate this effect. And with this, I would like to conclude. Well, I hope to give you some insights in how we observed sensory firing, sensory representation, especially with complex spikes in CRUS1 and to some extent also in CRUS2. Show that some cells have a very reliable encoding, others are more dynamic, can change depending on the context, it can also change dependent on the timing, especially for rhythmic processes. We found in that complex spikes were more related to input, were time-wise more correlated to input, simple spikes more to output. But in both cases, probably you don't really need them as a motor command, but more as a modulatory command. So this work was funded by Health Holland, so thanks for them. And I think the people that did most of the work have been mentioned. I would like to mention also Cheng and Luca, who just joined the lab as PhD students, and you for your attention.
Thank you, Lawrence. Thank you so much. Let me open up the discussions to see if there are any questions for Lawrence. We'll start the questions for you. I thought one of the most interesting things you described was how in the air puff condition, it produces synchronous complex spikes in response to the air puffs, less so with the other modalities. Can you comment about rate versus synchrony as these two different measures of how information might be coded in the Purkinje cell's response to the climate fiber?
Yeah, so thank you for that question. So it seems that there's also other work that we did and that I didn't present, that there is indeed a relation between the chance of evoking a complex spike and the strength of a stimulus. So that's probably not world shocking. And the air puff stimulus is a much stronger stimulus than the tactile stimuli that we applied, also because it's also associated with a clear sound, right? The mice can hear, not only feel, but also hear the airflow. So with that, there seems to be a linear relation, at least within boundaries, that the stronger the stimulus, the more complex spike we can trigger. But these come at the expense of the intertrial intervals. And so at least within a range of say 1 to or a quarter to 5 hertz of stimuli, we cannot increase or decrease the overall complex spike rate. And so and that is a quarter of a hertz is way below the resting firing rate. 5 hertz is much higher than the firing rate. So it's not that these cells are exhausted or so, that they cannot fire faster or cannot keep up with the ongoing thing, but it seems really that once a spike is fired, the cells strive to remain around the 1 hertz, so the 0.8 to 1.2 hertz that they normally fire at. And whatever we did, we cannot really increase it. We can only increase it by giving 10 hertz stimuli, which is really annoying to the mice. So they start really to get away from that stimulus. Then we could get a 10% increase in firing rate. I hope this answers the question.
Thank you so much. Yes. Are there any other questions from Lawrence? Lawrence, thank you so much for being with us. I much enjoyed it.
Okay.
Wonderful Tuesday.
Okay. Thank you all. And goodbye. Have a nice day.